regarding mechanism of evolution. So what is mechanism of evolution? Means how the evolution is been seen. So what is origin of this variation and how does the speciation will occur? Means for example, you have taken a petri dish of bacteria and you have put some antibiotic into that. After putting some antibiotic into that, you have seen most of the bacteria are eliminated, but some few bacteria are still living. So this you can call it as the variant, which is the variant that accommodated to the condition and that lived. Later it developed into a new species, which we call it as speciation. So even though Mendel has talked about inheritable factors influencing the phenotype, Darwin either ignored those observations or kept in silence. So he already told that Mendel talked about inheritance. Factors will be inherited from, but Darwin ignored these observations and kept silence. In the first decade of 20th century, Hugo de Veris, based on his work on evening primrose, brought forth the idea of mutation. So large difference arising suddenly in a population. So you have to remember here. So the Mendel work has been ignored by Darwin and the observation he kept silence, and later the scientist Hugo de Veris, he worked on evening primrose and he observed a mutation. That means large change has been seen suddenly in a population. He believed that the mutation is the cause of evolution. So mutation is the cause of evolution is been believed by Hugo de Veris, and not in a minor way, not the minor variation heritable that the Darwin talked about. Mutation are random. They are like directionless. You can't see a particular direction and it is very random. While Darwinian variations are small and direction. So Darwinian told in a directional way, but Hugo de Veris told in a non-directional way. So evolution for Darwin is gradual, while Hugo de Veris is believed mutation caused speciation. Hence, he called it as saltation. Means suddenly it will happen, and that too random and directionless. We call that thing as saltation. So Darwin told it is a slow process, and that too towards a direction. And Darwin neglected the work of Gregor John Mendel. What he told, it is transmittable. So single step of large mutation, we call it as saltation. So studies in population genetics later brought out some clarity. So when we studied about what are the genetical things that is present in population wise, then we came to know how some clarity. So let's talk about Hardy-Weinberg principle. So in this population, you can find out the frequency of occurrence of alleles of gene or a locus. For example, if you have capital P and if you have Q, so the occurrence of P and Q, P plus Q is equal to 1. This frequency is supposed to remain fixed and even remain same through generations, when same proportion will be there. Hardy-Weinberg principle is stated is using algebraic equations. This principle says that allelic frequencies in a population are stable. You can see this frequency is stable and it is constant from generations to generation. Means you will have constant number of species will be present, approximation of this type and that type. The gene pool, that is the total genes in their alleles in the population. The total genes in their alleles in the population, we call it as gene pool, remains constant. So this is called as genetic equilibrium, which means P plus Q is equal to 1. Sum total of allelic frequencies is 1. So now let's see the frequencies, the individual frequencies. For example, he named it as P and Q. In a diploid, P and Q represent the frequency of allele A and capital A and small a. So in Mendel we have seen capital A is dominant and small a recessive. Likewise in Hardy-Weinberg equilibrium, he substituted that capital A for P and small a for Q. The frequency of AA individuals in a population, he taken as p square. This is simply stated in another ways. Very simple. And the probability of an allele, capital A, with the frequency of p appear on the both the chromosomes. That is of diploid. Means simply like capital A, capital A. So he called it as p square. Okay. Similarly, small a, he alternatively written as q square. And small a and capital A is written as 2pq. So hence entirely like the occurrence of capital A plus capital A plus small a is equal to 1. He was written this thing as p square plus q square plus 2pq is equal to 1. Right? You can also write in another way. This is binomial expression. We commonly call it as p plus q whole square. The frequency measure differ from exact values. So the differences indicates the extent of evolutionary changes. Disturbances in genetic equilibrium. So if any evolution is happened, this genetic equilibrium will be changed. Means change in frequency of alleles in the population. So would then be interpreted, so there will be some change in this. So that may leads to evolution. So 5 factors known affecting the Hardy-Weinberg equilibrium. You have to remember these 5 factors. One is gene migration. Means going to the other places. Gene migration. Or you can also call it as gene flow. Genetic drift. Mutation. Gene migration, genetic drift, genetic recombination. And natural selection. These things only will affect the Hardy-Weinberg equilibrium. So genetic migration, genetic drift, genetic recombination, genetic migration, genetic drift, genetic recombination, and we have this natural selection and mutation. These are the 5 things will affect. So when migration of a section of population to another place, then the population occurs. Gene frequencies change in original as well as in the new population. So new genes and alleles are added to the new population and those are lost from old population. This would give the gene flow if this gene migration. This would be a gene flow if this gene migration happen multiple times. So if the same change could occur by chance, we call it as gene drift. Sometimes the change in allelic frequency is so different. In a new sample population, they could become a different species. The original drifted population become founders. And the effect we call it as founders effect. Okay. And next, microbial experiment shows the pre-existing advantageous mutation when selected will result in observation of new phenotypes. Over generations and generation, this would result in speciation. Natural selection is a process in which heritable variation enabling the better survival or enabling to reproduce and leave greater number of progeny. A critical analysis makes us believe that the variation due to mutation or variation due to recombination due to gametogenesis or due to gene flow or genetic drift, resulting in change in the frequency of genes, alleles in future generation. So, coupled to enhance the reproductive success, natural selection make it look like different population. So natural selection can lead to stabilization, which more individual acquire a mean character value. So directional change, more individuals acquire value other than the mean character value. Or disruption, more individuals are acquired peripheral characters at both the ends of a distribution curve. Okay, so this thing we call it as Hardy-Weinberg. So this is stabilizing. This is directional. This is disruptive. Right? So number of individuals with phenotype. So phenotypes are favored by natural selection. So you will be having all those mean average. Right? This is all about Hardy-Weinberg equilibrium.